I want to take a step back and talk a little bit about one of the major components of this, which is donor-to-donor variability. So, Michael, I'd like to address this to you first. All right, donor-to-donor variability, it's considered, to be one of, if not the primary source of complexity associated with establishing product comparability. So talk us through, why is this, and some maybe potential ways to even overcome that.
So I don't think personally we'll ever overcome this, at least in our lifetime, because we're talking about biological processes, right? When we talk about a small molecule starting material, throw it on a mass spec, you know exactly what it is, right? With starting material for cell therapy, be it tumor or apheresis, we don't know all aspects of that. We have no control over that patient material for an autologous product. And we understand in detail now that donor material used for characterization can yield very optimistic results compared to clinical experience. It's important for late-stage developers to evaluate their assets in earlier phases of clinical trials to better characterize real-world outcomes by eliminating the compounding variable of pretreatments. However, when you get to the clinic, you have pretreatment of the patient that can impact your product. This also kind of fits into your clinical strategy as well. So like if you take, say, a tumor and you're going to make a TIL, or apheresis, you want to make a CAR T, your clinical protocol should really outline how much pretreatment of that patient has occurred, right? Because the results could be very, very different. It's really important to build solid best practices for data management and product development as soon as possible. I said this probably 18 times, but this is an area where it's really important, where we compare our development experience with tumor or apheresis that's from donors to our clinical experience. And as we gain the body of knowledge, what does the real world say? Right? I remember an article, it was right after the Kite product got approved, where they did a comparison of the donor material versus the clinical material. It was like a 20% difference in process parameters. I've seen much larger than that in my experience. So we know that cell health plays a big role in growth kinetics and yield of our product. There are many characterization our product attributes that we characterize that are being evaluated across the industry to search for translational relationships between starting material and product quality. We just haven't found it yet. So we're constantly looking for this, but it's in terms of mechanism of action, not necessarily efficacy. We're looking at how does that starting material impact dose? So, there's a lot of knowledge being gained on this. I just don't think there's a definitive answer yet. And then another point that I think is important is with a TIL, you don't have there's no vector. But vector is another variable for your engineered T cells or cell products that could impact the process between donor material and real world patient material. So I don't think that the vector should be discounted here.
Yeah, sure. Very good point. Right. Okay. John, what's your perspective?
We can talk a little bit about vector change control too, because you can have multiple sources of vector and then you're back to square one for each one. So pick your vector and pick it early.
Right.
Don't change it.
Right. And I think if I had to select a word that described everything to be done in cell therapy and with regard to anything, it's always early.
Mm-hmm. One thing, John, though, that I always say to the industry when I give talks about starting material is you can compare it to like a simple salt solution. You have one molar sodium chloride, right? You order sodium chloride of a specific spec, you add WFI, you have one molar sodium chloride. So what if you couldn't determine the spec of that sodium chloride to any level of accuracy? What do? Lots and lots of many lots of 1 molar sodium chloride to characterize it. So the days of, we heard this when I started in the early 90s, 3 lots gets you a process validation. No, 3 lots doesn't get you there for cell therapy.
Sure. Thank you for all that information. That was really, really helpful. John, I'd like to hear from your perspective and from the CDMO perspective before we move on about donor-to-donor variability. And, why is it that it is the primary source of complexity? And
Yeah.
to Michael's point, is it, are we ever even going to come up with a solution? Probably not.
Well, I think Michael and we agree that it's, at least as is currently, as the field currently exists, it's never going to be perfect. It's just the heterogeneity of patients and their clinical journeys, if you will. It creates so much variability. Patient A comes in with 6 lines of prior, patient B comes in with 4. Each respond to those prior therapies differently. Some come in with higher disease burden, others do not. There are many, many different factors here. And I think it becomes very difficult to minimize donor-donor variability in the world of autologous cell therapies. What can you do? I think, I've already talked about previously standardizing things like apheresis across the industry in such a way that at least that variable is accommodated. And there are probably some other commonly used or commonly performed unit operations that could be standardized. Whether or not the field as a whole would be amenable to those, I think is very debatable. One thing we will see, and it's topical in the sense that over the weekend, BMS released some data around their collaboration with 2seventy Bio around Abecma. They're moving that trial into earlier stages, right? And so as we talk about the variability of starting material as it's related to lines of prior, you bring patients on board earlier in their patient journey, then theoretically, if we become frontline, I think that the variability will be less. Now, that said, is it ever going to be at a level where we want it to be? I don't think we're going to get there in autologous. In allo, and I think, somebody had asked a question, see if I can scroll quickly and just kind of answer this question. Michael Strauss had asked around addressing donor variability in allogeneic processes. The benefit of going allo here is that you're able to do that. And theoretically, you should be able to design, if you will, what you're looking for around some of these critical process parameters and the downstream CQAs. The caveat here, and I think this is where the field right now is currently answering questions, is what are the CQAs that are tied to clinical response? Once that question is answered, then I think what you're going to see is the allo field is going to leverage a lot of that biology to forward engineer some of those CQAs in their product and minimize on the front end the donor-donor variability as part of that process.
The one thing with allo, with selecting donor selection with allogeneic is a little less forgiving, I think, than for a standard cell, like a autologous cell therapy. And the reason is you can go through, genetic manipulations over space of years to develop your product, and then find that you didn't evaluate something in the donor, and now you're back to square one. We've seen this. There's some press releases I've read recently where this has happened at some companies. The challenge there is knowing what genetic markers are important and which ones aren't, and sometimes we don't know what genetic markers do. So, the allo question is more of donor selection for developing a product over a scale of a long period of time, whereas with autologous, it's collecting from a patient population that you're not really concerned about the same things you're concerned about with allo, with oncogenesis, et cetera. And the impact is smaller. It's one patient rather than an entire product. So interesting considerations. I think the allo is a little more difficult, to be honest.